this is a picture here of coronavirus. I'm sure all of you have seen this schematic of what the virus is, which is basically a strand of genetic material, which is the coil that you see over there. In the case of the coronavirus, what we have is an RNA coil, and I'll get into what an RNA and DNA is. The main difference is in the backbone of DNA versus RNA. DNA is deoxyribonucleic acid, and RNA is not deoxy, which means that there is an extra oxygen on RNA, which DNA does not have. And that oxygen exists in the hydroxyl group at the 2' position in the RNA backbone. This backbone attaches to different nucleotide bases, such as here you have a guanine nucleotide base, and RNA and DNA have basically the same bases, except for one of them is different. But it's a very basic language, pun intended, where you have G, C, A, U, and T as the letters of this language. And depending on which letter is in the sequence, it will encode for different proteins. Initially, different amino acids, which come together to form proteins, which is the building blocks of life. But really, the code of whatever form of life is going to get built is written in these base pairs and the sequence that comes out. And that's really the crux of how life, of our form of life, all form of life in today's world or in this world, where it came from. And that's nucleic acid bases, and both DNA and RNA are of that structure. So the question is, where did this originate from? In a paper that was published in the Proceedings of the National Academy of Sciences, we found that certain sugars such as riboses were found on meteorites. In fact, amino acids had already been found on meteorites before that paper was published. Now, that doesn't mean that that's where they came from, but that's one potential source. Another, there was an experiment done, which was published in Nature Chemistry, which showed that if you have a basic starting point of hydrogen sulfide and hydrogen cyanide, you can end up with organic molecules, including amino acids, including sugars such as the ribose, and even lipids. So you've got a single one of these rungs of the ladder form. And of course, they have an affinity to get stacked on top of each other because of the chemical nature of the compounds. But then the real question is, now this could have been just any other chemical. The real question is, how is it that a chemical was able to self-replicate? Because that is a signature of what life really is. And that is a mystery. There are certain forms of RNA called ribozymes, which are not proteins. They're still RNA with the same basic scaffold and structure, but a coding such that they become convoluted into a three-dimensional shape, which is able to act like a catalyst for enzymatic reactions. That's fancy words for the RNA was able and is able, by means of ribozymes, which is still a form of RNA, to replicate itself. We also have entities called viroids, which are circular RNA units, which have been seen to infect plants without the need for proteins. All these factors, as time progressed, helped bring forward this RNA world hypothesis, which was that the first self-replicating molecules were RNA strands. And that leads to the question of, is that life? Once you've gotten the ability to self-replicate. Because what viruses, such as the coronavirus, which is an RNA virus, the reason that they're not considered life is often that they don't have the ability to replicate in the absence of the host's machinery. Now, it's possible that viruses did have the ability to replicate at one point, but as they got dependent on host machinery, they lost that ability because that's the way they evolved. RNA itself is actually quite unstable. Because it has that extra oxygen group, which the deoxyribonucleic acid, the DNA, does not have, that oxygen moiety leaves it very susceptible to being damaged. And what that implies is that RNA is highly susceptible to mutation, and if you have a mutation which is positive, it's going to end up being selected. So, for example, let's say we have a situation where you have now an RNA strand which is able to self-replicate, and you can go from a single RNA to 2 RNAs, and through an exponential growth, you will have in each generation exponentially higher number of RNA molecules. So within, let's say, a few days or a few generations, you could easily reach 1 billion RNAs. And then, let's say the replication time is a day. The next day, you will have 2 billion. And before you know it, you're hitting trillions of entities of RNA strands. And then within one day, you're going to go from 1 trillion to 2 trillion. The point being that because RNA is unstable, there's going to be a lot of mutations in the trillion or 2 trillion RNAs that are found in the next day. Many of those mutations, predominantly, of course, the majority of those mutations are going to be deleterious. So they're not going to be helpful to help the RNA replicate any better. So many of those mutated subsequent RNAs may die off or may not be able to self-replicate further. However, maybe 1 in a trillion, 1 in 10 trillion or 20 trillion, which does not take very long to reach, molecules develops a mutation which is helpful. For example, it loses the hydroxyl group and turns from RNA into DNA, which is 100 times more stable than RNA. What's going to happen? Well, when you have a DNA molecule that ends up being found or formed, then the DNA is now going to be more likely to continue replicating because it was actually a more stable molecule than the original RNA itself. Let's say the same thing happens several generations of DNA later. You end up having one out of trillions of molecules, one DNA molecule develops a mutation which allows it to form a bond with another DNA molecule, such that it turns into a double-stranded DNA molecule, and which, of course, gives it further stability. And then the next thing that's going to happen is that double-stranded DNA is going to continue replicating because the original molecule had gained this mutation that gave it this propensity. This took a little bit of time to get into, but I wanted to be a little bit more thorough about it because there's a few basic concepts which are true for evolution even in animals today, or in evolution for larger animals too. One, that this is not a directed process. It's not like RNA saying, I want to become more stable, or DNA says, I want to become more stable. Because as we go into it, those things will come up. It's just that if a mutation arises which gives stability, it ends up being naturally selected. And therefore, it ends up being preserved. Now, sometimes there may be times where even positive mutations may die out because of random chance. But the random chances over billions of years are that whatever small, minuscule positive mutations may have occurred, some of them will get selected and will have an advantage in continuing replication. To illustrate this, let's talk about the history of the world from the very beginning, because the time scales are huge. Now, in this diagram, in order to kind of be able to show things, they've expanded the last 1 billion years, and the first 5 billion years are taking up lesser space than the last 1 billion years. So to rectify that, I've got sort of a table here with 5 squares representing each billion years of Earth's existence. So 4 billion years ago here, we have the formation of the first, what's called prokaryotic cell. Now, going back to the DNA, let's say the double-stranded DNA is replicating. If a mutation arises where it gets enveloped by a coat, and then that persists, that's going to be another layer of protection, which is going to help the DNA replicate further. And that's going to be a mutation that's going to be selected for. So we know that at some point before this 4 billion years ago, we had molecules of DNA or RNA which were replicating, and eventually they led to the formation of a genetic material which had an outer membrane, which was essentially the first cell, and the first cellular organisms were formed. And we have fossil evidence of groups of these cells existing approximately 4 billion years ago. If you had a situation where this genetic material was encased within the cell in a nucleus, that itself adds some selective advantages, because once you have genetic material which is further protected inside the nucleus of a cell, and you have some differentiation of functions within the cell, and that's called a eukaryotic cell. But once you have these division of labor, then that gets selected for, because that allows you for more specialized functions, and that second development actually took an entire 2 billion years just to get to a point where you have a eukaryotic cell, which is that the nucleus is encased in a layer and you have some division of labor within the cell. And one theoretical possibility again is there that viruses may have been involved in that process, where maybe one of these simple cells was infected by a virus and the genetic material got encased inside, or maybe one cell engulfed the other and genetic material stayed inside, and a selected mutation arose, which allowed for this process to continue. But the point being is that if you go back 2 billion years ago, the only thing that developed in the 2 billion years, which is half the history of life, was that you went from a single DNA strand to in a envelope, a simple prokaryotic cell, to a eukaryotic cell. Another billion years, go halfway across until even now, and the next mutation, or the only development that ends up happening, is that these cells figure out a way of clumping together to form organisms that are called sponges. And again, that's a very simple development, but what this is showing is that it's taken immense swaths of time to develop very simple mutations. And so you end up going until just 1 billion years ago, the development of what are called sponges. And then the rest of complex life starts developing approximately 500 million years ago. You have what's called the Cambrian explosion, where there's a lot more diversity that develops. And literally, human beings only have shown up in the last few seconds if you go through a 24-hour cycle in the history of life. What ties this entire story together is what we're dealing with right now with coronavirus. Why? Because coronavirus is a single-stranded RNA, which doesn't replicate anymore on its own. The machinery that the RNA is using to replicate itself is something that we possess. And why do we possess that machinery? Because there's a link going back all the way, possibly, to the very beginning of life. The fact that this virus recognizes receptors in our body, the story of how this virus has come to be, is a story of life itself, and it's based in the realities of biology that tie us back to the very nature of life itself. Thank you for watching, and I'll see you next time.